Now, proteins get a little complicated here. Um, Proteins can have many, many, many different structures and functions, and they're actually the direct result of gene expression that we'll talk about later. Now, a protein monomer, as we've talked about already, is an amino acid, and it's composed of an amino group and a carboxyl group on either side of a alpha carbon. Now, sticking off of this alpha carbon is then a side chain, which could be any number of different functional groups. There are actually about 20 different varieties of functional groups that you find that are attached to these alpha carbons, resulting in 20 kind of unique different amino acids. Now, polymers of amino acids are called polypeptides, or maybe a protein, but we'll get to that in a little bit. Now, if it's not apparent to you already, you should definitely know the structure of an amino acid. I've mentioned it like 5 times at this point, so definitely know the structure of an amino acid. Okay, so let's dig into the polymers of amino acids. Well, I mentioned they're called polypeptides, and that's because the things that link these amino acids together are a series of peptide bonds, and they are between the hydrogen in the amine group and the OH of the carboxyl group. Do a little dehydration synthesis, and bada bing, bada boom, you got yourself a polypeptide. Okay. So again, the reason they're called polypeptides is because right here is that peptide bond linking the amine and the carboxyl of the adjacent amino acids. Now, to kind of orient yourself on polypeptides, look at you can either look at the N-terminus, which is where the the amine is, or you can look at the C-terminus, which is where the carboxyl end is. Why would you necessarily refer to this as either the H or the OH side? Well, because remember, you lose the H or the OH in those dehydration reactions. Now, before this chain is called a protein, you actually need to do a bunch of kind of extra steps to modify its structure. And we kind of summarize that as folding. So you need to do all this folding of this amino acid chain to form this final kind of 3-dimensional structure that makes it a protein, really. So we talk about the different kind of hierarchy of protein structure, calling it primary or secondary or tertiary or quaternary. So in other words, there are 4 levels of structure that occur. The first one here, primary structure, is basically the kind of what we already talked about. It's this series, the particular sequence of amino acids joined together by peptide bonds in a polypeptide chain. So the particular order of amino acids is unique to each protein. Okay? And this order is actually determined by DNA. We'll talk about that very explicitly later on when we get to gene expression. Now, secondary structure is now you have this kind of chain of amino acids with its R group sticking off. These, the bonds will actually occur between the aminos and the carboxyl groups. So you'll have hydrogen bonding that occurs between these kind of backbones, right? And so this hydrogen bonding can result in either alpha helixes, like you see here, where they kind of spiral around each other, where you start seeing the hydrogens and the oxygens of the amines and carboxyls of the chain Right, so you have this long chain here, and like you have the hydrogen and the oxygen. They'll start kind of interacting like this, and they'll start forming those alpha helixes where it's a kind of a spiral, or this beta pleated sheet where you cause kind of like ribbing along the chain. And again, this is only between the aminos, or the amino groups, and the carboxyl groups. That's where this secondary structure is derived from. Hydrogen bonding between the aminos and the carboxyls. The R groups are not involved in this secondary structure yet. Or they're not involved in the structure at all yet, really. Okay, now in the third or tertiary structures, this is where the R groups actually start interacting in all kinds of crazy ways. So you can have ionic bonds between the R groups. You can have van der Waal interactions. You can have hydrogen bonds, disulfide bridges. It does all kinds of crazy stuff to really kind of twist and turn this protein in a very convoluted and complicated shapes. That's because you have all these different R groups kind of sticking off the side now that are now starting to interact. It's very complicated. Now, finally, you can have quaternary structures where you actually have several of these kind of the chains that you started out with that are then folded and then, curved based on the functional groups. So folded based on the amino acid, or the amino and carboxyl interactions, and then folded again from the all the R groups. Now you take a couple of those and you stick them together, and all those R groups start having interactions. You have hydrogen bonds between the amino acids, bonds between the R groups, and then all these folded polypeptide chains result in a huge number of kind of unique combinations of proteins and structural shapes that can be vastly complicated, but also theoretically totally predictable, right? So you could predict what kind of interactions you'd expect between the different functional groups or the different amino groups, right? There's actually this really interesting software you can go and check it out called Foldit, where you can go online and they've more or less crowdsourced, identifying protein structures. So scientists have put it out here. It's turned into a game basically where you can go in and play around with these 3D models of proteins and try to determine exactly the right shape of these different proteins. And they've actually very successfully identified protein structures that they had no idea what they were before until kind of crowdsourced the science here. It's pretty interesting. Okay. So figure 3.3 is a pretty nice summary for all the different layers of protein structure, from the primary protein structure of the chain of amino acids to the alpha helixes and beta pleated sheets of the amino and carboxyl interactions, to the R group interactions and how that bends and changes it. And then finally to how what happens when you start mashing these bent up folded structures together to form this final quaternary protein structures. Okay. Whew. So I spent all that time talking about how to get these very complicated, vast structures. But of course, you can also lose those structures. Now, denaturation is the loss of a protein's secondary, tertiary, or quaternary level structures. Now, this usually happens or could happen from several different factors. Typically speaking, a big change in pH or swings in salt concentration, or very likely huge increases in temperature will cause these bonds to break. Typically speaking, you usually see the third and fourth level structures break before the secondary structures. But hypothetically, if you got things even hot enough, you could actually break the primary structures of proteins as well, but they'd have to be pretty darn hot. So that's kind of the idea, is that you can increase the temperature or change the pH dramatically or adjust the salt concentration dramatically, and it'll cause these different structures and these different bonds to break down and eventually denature and break apart. Now the real question is, for you guys, what happens when you do this? What happens when you denature a protein? What do you think? Well, you're fundamentally changing its structure, right? It's losing its structure now. So that probably means, right, it stops functioning in the same way. It stops being active. It becomes biologically inert, in other words. So you can actually have processes that redo those tertiary and quaternary structures, particularly, right? But it doesn't necessarily always happen. So yeah, so that's what protein denaturation is. Okay, I know we've been at this for a little while now, but really, really fast, let's do nucleic acids. Now, nucleic acids, the monomers of nucleic acids are called nucleotides. And there are kind of two broad classes of nucleic acids. You can have deoxyribonucleic acid, or you can have ribonucleic acid. Now, these are primarily involved in things like heredity, and they can also help determine, they also determine what proteins get produced. But we're going to talk a lot more about these later on when we get to genetics. So don't sweat them too much right now, other than understand the monomers and nucleotides, and you can have either DNA or RNA, and they're generally involved in heredity and protein production later on. That's it. Okay, before we move on to lecture 4.1, where we'll talk about the process of abiogenesis, which is pretty interesting. We're going to do lecture question 3.2. Which of the following are examples of polymers? Amino acids, phospholipids, polypeptides, or proteins? So you need to identify which of those are polymers. How can you do that? Well, you could try making a table that looks a lot like this. So I really recommend you guys go ahead and do that. Put together a table, and you can fill out pretty much all of this, except for, I don't know how much of the nucleic acids you can fill out. We only kind of briefly dived into that. But you should be able to definitely fill out the carbohydrates, lipids, and proteins portion of this table. And I'd recommend doing that before next lecture. See if you can come up with some great everyday examples. That's also a good option. Okay, that's it for lecture 3.2. We'll see you in lecture 4.1, where we talk about abiogenesis. Bye. Oh, wait, no, not bye. Just kidding. I've been lazy and haven't done my BIOS form at all. But that's okay. You guys should be pretty comfortable at this point. But just in case you're not, we can go through it real quick together. Should look something like this. Right? So you have the importance of carbon. You have intro to carbon, hydrocarbons, functional groups. So that was all content we talked about in lecture 3.1. And then we have biological macromolecules, carbohydrates, lipids, proteins, nucleic acids, and also stuff we just talked about just now in lecture 3.2. So, but I'm sure you guys had that already, right? You're super comfortable with BIOS forms at this point, I'm sure. You're old hats at this, by now. Anyway, see you in lecture 4.1. Bye. Oh, hey, before we leave, though, a quick note about lecture 4.1. So we're going to be talking about the process of abiogenesis in that lecture. However, it's worth noting that you cannot actually find that content in the book, in the OpenStax book that you guys have been, referring to up until this point. So really, that means you guys don't have to worry about skimming the content ahead of time. It's, maybe missing out a little bit of preparation, but that's okay. Just stick to what we talk about in the lecture. If you really want to get into it, we are going to be using some of the figures from the Campbell. So that's 10th edition. This is what we used last year and the past few years that we've gotten rid of. So Campbell still talks about this stuff, but OpenStax does not. But we really think it's very important that you guys learn about abiogenesis because it really sets you up for what we talk about in Gen Bio 116. So the figures you're going to see in lecture 4, so 4.1 and 4.2, they're all going to be from the Campbell book. So if you see like figure 25 point whatever, if you Google Campbell 10th edition figure 25 whatever, you'll find those figures. That said, honestly, I wouldn't worry about it. Just watch the lectures, and that's probably going to be good enough for you for lecture 4, so 4.1 and 4.2 for me. So just a heads up, don't sweat if you're like frantically hunting down, trying to find the content in OpenStax, like where's abiogenesis? I don't understand. Don't worry about it. Just watch the lectures. It's cool. It's all good. Okay. See you in lecture 4.1.